The overall aim of this procedure is to demonstrate a method of monitoring the calcium signals at both the cellular and whole plant level. This is accomplished by first growing seedlings on the MS agar plate or directly within the imaging chamber. The second step is to transfer the seedlings onto an adhesive film or to keep them in the imaging chamber. Next, stimuli are applied to the seedlings to induce calcium signals. Ultimately, live cell luminescence or fluorescence imaging is used to show whole seedling or cytosolic nuclear calcium response respectively to different stimuli.
The main advantage of this technique over existing methods like single plant aquarium assay is that film adhesive seedlings assay is simple, sensitive, and robust. Therefore, this technique can be adapted to the high throughput screening for mutants with altered calcium signal responses. In addition, compared to FRAP-based calcium imaging, using GFP-based genetically encoded calcium indicator avoids complicated microscopic settings and calculations.
This method provides insight into calcium signaling during the stress response in Arabidopsis, but it can also be used in other contexts, such as during development or defense in other plant models.
Demonstrating this method will be Shenyu, a student from our lab.
To begin this procedure, sterilize the seeds of Arabidopsis plants expressing Aequorin with 10% bleach solution, containing 0.01% Triton 100.